Some of the scientists is they can't hear me. Is that? Yeah, I know. God damn it. Hello, everyone. Welcome to the Magic Sandwich Show. I'm your idiotic host, JD Kane, who doesn't know how microphones work. Sorry. So start 3, 2, 1. All right. So welcome to the Magic Sandwich Show. Tonight we're discussing mutations, evolution, and maybe some other stuff, but mostly about mutations. Yeah, shut up, Crispr. Well, let's get started. Let me introduce who's on tonight. For those of you who didn't read the title, First, we have Cameron James. Cameron James is a retired detective and a big fan of, well, these discussions, creation and evolution. So, Cameron, can you tell us a little bit about yourself?
Well, I had a job where people were dying to meet me. And anyway, now I'm just I like science, and I think of like Michael Faraday, who invented electricity, and he was a bookbinder and would read books at night, and he liked books and science, and he found out more about electricity than anyone else at the time. So I tried to do the best I can. I'm not going to be an expert, but detective, they kind of look at the big picture and try to make a case from the big picture. And you have experts in different areas, but I think it's important that we have somebody look at the big picture, and that's what I try to do. And let's see, I don't know how much time I got. I can talk for a while here, but yeah, I'm a retired detective. And anyway, and I hope we have a good time discussing things tonight.
Hey, maybe you can write a book, Cold Case Christianity 2. All right, so
I have written a book.
Yeah, that's nice. Well, wait, we'll talk about that later, actually. I'm actually curious about that. I want to hold on to that. I want to know what that book is about. So Crispr, I've known you for a while now. Tell us a little about yourself.
Sure. So I've actually been off and on part of the community here since about 2006, I think. So I probably predate a lot of people. But I haven't been terribly active until a couple of years ago. My background is in the sciences, specifically biology. Currently working on my PhD in biology, so I have some familiarity with the topics. So,
all right then. So, like I said, tonight's topic is about mutations. Now, I'll be honest, I'm not entirely sure where this is going to go. It's kind of an odd, kind of general topic, mutations. Like genetic ones, we talk about the X-Men. I mean, this could go anywhere. You just never know. Like, is anyone going to learn how to fly or steal people's abilities? I want laser eyes. I don't really want the claws. They look like they hurt.
The claws weren't a mutation.
Oh, shush. Well, no, he had bone ones to start.
Depends on which source material you're looking at.
Damn fans. All right. All right. By the way, for those of you outside who are wondering why we're not getting, 3,000 viewers again, someone, not saying who, may have set the time to be later than it was meant to be. Not saying who. So we're actually a half hour late starting, while the other people think it's a half hour early. So who knows? Maybe in 30 minutes, everyone's like, "Hey, wait, the show's already going? You guys suck." So we'll see how that plays out. Anyway, when we start, Cameron, when we start with you, what is the? I mean, this is kind of partly your topic. What did you want to talk about when it comes to mutations and, I guess, evolution?
Well, I want to talk about what detectives do, and basically it's what philosophers do. And I think all scientists should be good philosophers. And there's 3 tests for philosophical truth. And first is empirical adequacy, which means basically there has to be evidence that shows that this truth is valid. The second is experiential relevancy, and that is basically with science, it's doing tests to validate it and what you've experienced in life and different things. And I do this with detective work and murder cases, but the final one is logical consistency. So it has to add up, it has to make sense, and it can be validated with 1 + 1 = 2 mathematics, and if it's 1 + 1 = 3, you know it's wrong. And I see that more as a way to prove something wrong than to prove something right. And I think a lot of people in science today are using mathematics to validate things rather than empirical evidence. And I'm a big empirical evidence guy. And talk about pseudoscience doesn't use good philosophy. It starts with a conclusion. It's not good detective work, and they work backwards trying to validate. And in the process, they make grandiose claims that go beyond the evidence and just look for favorable evidence and ignoring other evidence. But real science follows the evidence wherever it leads, and claims are conservative and tentative when you come to a conclusion, and you have to be able to consider all the evidence and follow valid logic, of course, and change when new evidence shows you wrong. So if you got an idea like, oh, I know JD killed his wife and his wife's dead, so I'm just going to look for evidence that JD did it, and JD might have, well, look at this, look at this, look at this. Like, I don't care about the other evidence. I want to convict you, you know? And so that's wrong type of thinking, and I think that's bad science. And anyway, in looking at mutations, the original idea of Darwinian evolution was that the change power was pangenesis, and it was basically Lamarckian methodology. And if you're familiar with that, but he called it pangenesis in his book. I think he's got a chapter on it. And what pangenesis does is supposedly is that, say, a giraffe can't reach the tall branches of a tree, and it has a desire to reach them, and then it changes down to their genome, and the offspring will have a longer neck because of that. And Darwin got most of his ideas from his grandfather, and his grandfather got them from the Greek philosophers, and it goes way back. But anyway, the story wasn't a new idea, and he validated his story with pangenesis. So as the power behind it, he had the big influence. Of course, the title was natural selection, but I believe in natural selection, but I just don't believe natural selection has any power in itself. And so anyway, in the 1890s, they start figuring out mutations, and in 1930s, there was a book written by R.A. Fisher, and it was the genetic theory of natural selection. And so that started a new push to change pangenesis to mutations. And at the time, they really didn't understand that much. And eventually in the 1940s, with the help of the Royal Society and Huxley and Mayer and different ones, they got neo-Darwinian evolution, which is the power of change is mutations. So that's basic stuff that I want to start out with. We'll let Chris do some, maybe.
Well, I mean, I don't want CRISPR just like try and correct anything. So what is the main thrust? If you have an argument, what is the main thrust of it? That mutations don't drive, or well, I guess that natural selection and mutations don't drive evolution, or that mutations are non-random? I mean, do you believe mutations?
Random. Yeah, well, my basic premise, axiom, is that mutations are they're basically an accidental copying error in the DNA RNA transcription copying process that leads to structural change. I think we can agree on that. That's probably from Wikipedia or something like that, I think. So I'm just saying that, well, even in mapping the human genome, they categorize mutations as either deleterious or functional. None are considered to be beneficial. So I would call them that they're structural changes that do not add any new function or new novel information that has a purpose. And so there's different types of mutations that we can talk about, and it's important that you understand these. And Chris, let me know if I make a mistake. And there's deletion and insertion in the chromosome. And then there's duplication, which repeats a segment in the copying process, and an inversion where it gets flipped over within it, and then a translocation where it's moved to a whole different area. But with the mutation process with neo-Darwinian evolution, they believe that mutations, it has to be the point, removes a chromosome, an ATCG, and puts in a different letter, and that makes a change. And so that's a possibility, but in order to make a major change, that's going to be really hard to have two mutations at the same time do two different deletions and insertions, and then have it result in something. And the odds of that happening in a mutation, first of all, with had them two right next to each other, it's supposed to be 1 in 14 trillion or something like that, or trillion or whatever. So it's not real likely. And anyway, so just to have that change process for something positive, I think is just not realistic to have it be strong enough to move something forward. I think if you had two together, maybe you could have a slight bend in your eyelashes or something like that. But anyway, I think that the DNA copying process is amazing. You know, that DNA is amazing. In our bodies, we have, well, I don't know how many. It used to be 30 trillion cells. Now they're saying 70 trillion. I heard somebody say 100 trillion. We got trillions of cells in our body, and every day there's 50 to 70 billion cells that die. And so we have this copying process going on, and DNA is truly amazing. But they say DNA in your body, they used to say if you ran it, stretched it all end to end, just in one human body, it'd run back and forth to the moon over 100 times. Now they say twice the diameter of the solar system. And that sounds like more than 100 times back and forth to the moon, but your DNA is truly amazing. And so what happens is that your cells are amazing, and when they're having to copy all these cells, the first thing they do is process the DNA, and it involves a process that's called mitosis, and it involves an army of tiny molecular machines. I think they're electronic, but that'll be a different argument for a different time. But anyway, they're amazing, and you can see on YouTube, and they just what all there is involved in copying this. It's hooks up to a helicase is what it's called. I think it's like a helicopter. It spins it faster than a jet engine, and then it has a little thing that comes and separates the DNA, and then it makes copies of the different strains, and then the strains are amazing in themselves. I think there are 3 million different proteins in it, and then the problem is the program runs one way on the bottom strip, and then it runs the opposite way in the top strip. And then so they have to be taken apart, and then they make copies, and they send this one around. They got to flip it around and attach them. And the DNA and the RNA little molecules are coming, and it's really amazing. And once it's finally put together, then if there's any mistake made, the helicase will stop immediately, and they'll fix it. But supposedly, let's see, if there's some mutations in there, there's an army of two different types of different machines that go over it and will fix all the problems in the DNA. So just the cell itself is amazing. The process goes on, and the proteins is a tangled mess, and they wrap around proteins, histones, and then they come nucleosomes, and then they all get together and become chromosomes. And so anyway, it's really an amazing process, and just how this all started and how it all got that way, I believe is a big problem. And one other thing I'd like to cover is you have these different types of mutations. There's something that they call a back mutation, and This is not really what I would call a mutation, and this is defined as a mutation that reverses the effect of a previous one, and could at one stroke revive a complex function that had been shut off earlier, and but it turns on something that's already existing, the existing information. So with a back mutation, which I believe that's how the finches change their beaks, and they're not random. They seem to be direct response to what's going on. So maybe we should go back to Lamarckian principle, or else it was programmed that way originally. And so the finch, they get to a harder nut to crack, and so then this back mutation will cause the offspring to have harder beaks, and their beaks go back and forth, and depending on what's going on. And I think I compare this to like a car. You can say, well, you drive up a mountain, your car's going to shift gears. Was that caused by the mountain, or was it designed to shift gears? So I'm saying that all this cell and everything points to an intelligent designer. And anyway, see what Crisp has to say. I don't want to take all the time.
No, that's good. I mean, that gave us. We will definitely fill the time with that. So thank you for the spiel.
Okay.
Crispr, unfortunately, has already drunk like 4 bourbons now during that speech, so we're not going to get anything useful out of him at this point. Just going to be slurs, and he might throw a glass at you. All right.
No, I don't slur.
I didn't slur. Yeah. All right. Hey, that's true.
I'm going to go get a Michelob, okay? Just kidding.
I was tempted, but I had to wait till you were done. All right, so CRISPR, I mean, that's an awful lot to deal with.
Well,
I'm not going to demand you deal with all of it at once. So just pick whatever you.
We'll start at the end because it's the freshest in my mind. So you started talking about back mutations in the Darwin's finches. Is there evidence of back mutations in those finches?
I kind of understand back mutations, and I think that's the best explanation for them.
Yeah, I'm just asking a direct question.
I haven't validated that by Google yet.
Okay, so my understanding is that there's no actual evidence of any back mutations per se in those finches, that it's predominantly a question of allele variation, right? So it's
just that they can transfer back and forth. It shows me that there's something else going on.
Well, that's not a back mutation.
Okay.
A back mutation is when you have a mutation occur, right? And then the reverse mutation occurs later on in the lineage.
Okay.
So, right. So what I'm trying to point out here is that you just brought this thing out of Darwin's finches have back mutations.
Well, that's kind of a guess.
As soon as, okay. Well, okay. So, but I don't know why you're making a guess about this, right? This is the kind of thing you want to research and then talk about on the basis of the research. You talk about being a detective. I mean, yes. do you just make guesses in court? Like I have no idea, but I'm gonna guess. Like no, you've got to get the data first, right?
Uh-huh.
Well, I think that's a good policy, right? So I would suggest probably let's try to avoid introducing conjecture into this, right? Because that's just going to confuse things and overcomplicate and distract.
Do you think they're done by mutations?
I think that a great deal of the source of variation is from novel mutation. Certainly, there is reasonable evidence to that effect. When they've done studies, they've found they've been able to isolate novel SNPs, SNPs, single nucleotide polymorphisms within the different populations that seem to assort with the different phenotypes that are observed. Mm-hmm. So that would be evidence of mutation being involved with the novel phenotypes. But there's also a great deal of existing phenotypes changing in abundance to produce that is to say, the existing alleles change in abundance. to change the phenotypes that are expressed. So there's some of both, but there's definitely evidence of novel mutations in those populations.
Well, new information that wasn't there. It's just a matter of plugging an ACTG in a different slot and call that really, I guess you could call it novel, but I don't think it's purposeful.
Well, so that's sort of putting the cart before the horse, isn't it? Right. So if you want to say that there's some novel bit of function, right, the first thing you have to do is identify novelty. Right? Let's find anything that's different between these two populations. Okay. And then you ask the question, is any of this actually doing anything? But firstly, there is demonstrably heritable variation in the form of these SNPs that correlates with differences in phenotypes. Now, that's pretty strongly suggestive. If you talk about circumstantial evidence, that would be very strong circumstantial evidence that these are in some way causative.
Okay. And so, is that really strong enough for the type of change that Darwinian evolution requires?
Well, what is the type of change that Darwinian evolution requires?
Well, you go from a amoeba to a whale, or amoeba to a human.
Not in one shot.
Well, experientially, maybe they should test maybe bacteria and see whether they can get.
So, how many? Let's think about it this way: how many generations are there between, under the current model, between the amoeba and the whale? Not an exact number, just ballpark it in terms of a few or a lot.
Well, I think there couldn't be enough for it to work.
Well, that's not the question.
So his answer would be a lot, right? There'd be a lot. A whole lot. Okay.
In other words, if think about it this way, if you were to go ask some professional biologist, how many generations were there between the last common ancestor of all eukaryotes and whale, right?
Uh-huh.
Do you think that they'd give you a number larger than 1,000?
Yes.
Maybe larger than 100,000? Probably, it's going to be a big number, right?
Yeah,
that's the point.
A big number.
So in other words, the change that Darwinian evolution requires, the kind of change it requires, is 1,000th or 1,000,000th or 1,000,000,000th of the difference between these types of organism, right? It's not moving from an amoeba to a whale, right? It's moving from an amoeba to something like kind of like a slime mold, maybe, and from something like that to something kind of like a sponge, and from something like a sponge to, and so on and so forth, right? So, whenever you present the question as an amoeba to a whale, that's sort of misrepresenting the scale.
Okay, so what are some examples of functional beneficial mutations that I Googled that and didn't really find much?
Well, how are you defining beneficial?
Well, there's different ways, but can come up with a new function, basically. I know that they have one is bacterial antibiotic resistant bacteria, but I wouldn't include that because the ones that are able to resist are they survive, but they're not as viable as the ones that are killed by bacteria, basically. So
well, they're more viable in that environment, but that's not really the point. Yeah. So let me ask you this: if there was a mutation that, say, modestly affected the length of the long bone of the leg. Okay? In humans. Let's just say in humans. Would you call that as beneficial, neutral, deleterious? What?
Well, I don't know. Some children are taller than their parents, and it all depends how what you're trying to do. And maybe if you're an astronaut and you're 6'2", you're too big to fit in a space suit, and so that's not advantageous.
Okay, so everything you just said is exactly the point of why I asked you what you meant by beneficial, because the majority of mutations that have some effect on phenotype have a very small effect on phenotype. And a small effect on phenotype is very likely to be very situational in terms of its effect on fitness. So taking this example of an effect on the length of a long bone in humans, which is, by the way, a real example. They've actually identified this gene. I don't remember the symbol offhand, but I can find it later. You can imagine quite readily environments where being a little taller or a little shorter doesn't matter. And you can also imagine environments where being a little taller is great and being shorter is bad, or where being shorter is great and being taller is bad. Or environments where you really need to be that middle height, right? You can imagine all of these possible environments, and it depends on which one you're in, whether or not this particular gene, this particular allele for this gene is actually beneficial. But so, clearly, clearly in some environment, it is beneficial.
But it really doesn't change it to anything but a bone, and it stays as a human bone. And to get an arm to turn into a wing is going to be another big problem.
Okay. Can you point to the bit of the wing that is fully novel with respect to the vertebrate arm?
Well, the feathers and the aerodynamic, and just if there was a mutation that caused them to not have a claw but have a wing, I don't think they'd survive.
Well, there's certainly no mutation that ever caused something to replace a claw with a wing. I don't think anybody's claiming that, though.
Well, in the process, it would cause them to be eliminated, I would think.
Yes, but that's not the suggestion. Nobody is suggesting that the course of evolution for birds' wings was to replace a claw, which is a distal portion of a vertebrate limb, right? The very, very tip, with an entire wing, right? Firstly, because that's not what the homology shows, right? If you look at the homology of a bird wing, right, you can clearly see all of the homologous parts to the to our arm, right? Mm-hmm. You can see how everything works in a one-to-one relationship.
It takes a good imagination.
No, it doesn't, because they're the same bones. They're produced in the same order by the same genes in the same pathway.
Okay. And so anyway, let's take the example that comes up, and it's lactose tolerance in certain individuals. And anyway, they say that's a beneficial mutation. And basically what happens is that apparently in the past, humans, when you have enzymes to digest lactose milk, and it turns off at a certain age, and then some people still have that, and some people, Europeans, can still, it doesn't turn off, and so they can drink milk their entire lives. So people say, well, that proves that mutations are good. Well, it just proves that the thing that was supposed to turn off doesn't work anymore, and so it's not really gotten any better. And Asians who are not able to drink milk, they haven't been affected health-wise that I can see experiential, or they haven't been inferior intellectually because they haven't had milk. So I don't think that's a good mutation.
So can I jump in on that?
Yes.
Right. So the fitness benefit is if you live in a culture that has ready access to milk, right? Cow's milk is a great source of nutrient, and if you're able as a child or an adult even to drink it, right? That's a huge benefit. Okay. And in order to take advantage of that, you need lactase persistence. Okay. So when you have a mutation that causes the persistence of lactase expression, right, which is the thing that lets you tolerate milk, that is beneficial, but it's only beneficial in those environments where milk is plentiful. So if you go to China, right, 8,000 years ago, and they didn't have a lot of cows, they weren't producing lots of milk. They had no benefit then to have lactase persistence because they didn't have the milk resources to take advantage of with it, as opposed to the European ancestors who did have cows. They did keep livestock of that sort, which is why it was beneficial for them, but not for the Asian population.
And I see that by design. And I think like the color of our skin, light-skinned people, if they're in the sun too much, they get different kind of skin cancer. They get eye cancer, and they have a lot of problems. But darker skinned people, if they go up in areas like Europe where there's not as much sun, they don't produce enough vitamin D3. And so I think it's by design and maybe by mutation or whatever, but I think it's by design so we could go around the world is the way it was. And it came from the intelligent designer who was the first cause of everything.
So here's the thought. If I were an intelligent designer of humans, right?
Yes.
I would design the genes that control the expression of eumelanin, right? To just turn on under prolonged exposure to the sun, right? So basically, if you had a European out in the sun all day for a long period of time, he would just start producing eumelanin, right? And his skin would become very, very dark, not tanned, right?
Mm-hmm.
But it would take on the same complexion that people from equatorial regions have, right? But that's not what we observe. Instead, the European is never
bad design, but you're saying that's a design. So I guess we.
No, I'm saying that this is the way I would do it if I were intelligently designing it, and I can imagine the regulatory pathways that would control for such a thing. They actually would not be difficult, okay? But instead, what we have is I as a European, if I want to live in the equatorial region, I need to douse myself in sunscreen every time I walk out the door.
Yes.
Right.
Yeah,
well, that's not a particularly great design, is it? Right.
Well,
because you could have just one little change, right, and everybody would be equally adapted to live everywhere.
Yeah, that would be ideal, but that's not the way it is. And to say it's a bad design when it's also incredibly complicated and there would have to be kind of a back mutation type thing in order for it to do it. And
why would it have to be a back mutation?
Because it'd have to switch back and forth.
Nope, nothing switches back and forth. The expression turns on or off, but the same genetic controls are in place the entire time.
If I may, I have a quick question. Cameron, do you believe complexity is evidence of design?
Well, I think, if it's functional complexity, and I mean like a ice crystal or a snowflake or whatever, that is a evidence of a design. And I think it's because it was originally came from water that had this design program into it. But I would say that that's not evidence of a designer because it doesn't have any functionality to it. And if it was full of little micro machines that were running back and forth and copying new cells and doing all this different thing, then I would say that it's probably came from an intelligent designer.
Okay, all right. You guys can continue.
Okay, I got another question for Chris. I like his answers. So anyway, there was a Nobel Prize winning grandfather of modern human genetics, which was Hermann Mueller. He said in 1850 that we could handle a load of about 0.3 mutations per individual per generation, and that was the limit of human mutational tolerance. In 1971, Nobel Prize winner Manfred Egan also calculated that the maximum number of mutations allowable per generation was 1 per genome per generation, and he said any figure above that would eventually result in genetic error catastrophe, a term he coined. So anyway, now we know that the Genome Matching Project found that there are between 100 and 300 mutations each generation. So were those guys way off line, or were they right? And it was is it a catastrophic error like Egan thought, or what do you think?
So there are several issues with this. The primary issue is that when this was initially discussed, people were thinking in terms of simpler genomes that have extremely compact levels of functionality, right? So if you think about, for instance, a bacterial genome, right? It's basically straight through with coding and regulatory, right? There's very little slack in there.
They just didn't have the knowledge.
Right. So eukaryotic genomes, especially higher vertebrates, higher animals, tend to have quite a lot of extra genetic material. Now, this is called the C-value paradox. There are a number of thoughts as to what's going on there, but the major idea is that in these more slowly replicating animals, it's necessary to have a bunch of space, okay? Because this both allows for recombination to work more effectively, and also serves as sort of a protective role against the active mutations. And so when you factor in the fact that only about maybe 10% of the genome is actually under strong conservation, which is the best evidence of functionality, then things start to make sense.
Epigenetics, it doesn't mutate though, does it?
No, it does. So this is people often get confused with regards to epigenetics. Firstly, a lot of people hear epigenetics and I think that they think magic. But really, anytime most epigenetics is not inherited is the first thing. This is very important. People always like to throw their attention onto these rare instances where epigenetic modifications are actually inherited. That is the exception and not the rule. But every time you see any kind of epigenetic regulatory system, that system is based ultimately in the genetic code. And that means that it is under the same evolutionary forces as everything else, right? Because if you have a CPG island, right, that means that you literally have CPG in the genome, right? And if you add or subtract the number of CPG sites, but by way of standard mutation, you're affecting the extent to which epigenetics can occur. So yes, epigenetics is absolutely amenable to mutation.
Epigenetics tells the cells whether it's a brain cell or a lung cell or a skin cell or whatever, right?
That is something of an oversimplification, but I think I take your point.
Yeah, it kind of gives it the information. There's something interesting that they in the mapping the human genome, they found that there's 3.6 million nucleotide mutations in the average human today. And what they found was they found an Eskimo that they dated that was frozen alive 4,000 years ago, and he was a male paleo, and he had about 450,000 nucleotide mutations. So 3.6 million and 4,000 years ago, 4,500. It appears that things are deteriorating and in the human genome. And
Can I ask where your source for this is?
Oh, you can Google it, I guess. I should have got it to JD, but it is a study that was recently done.
I mean, that's not nearly enough information to track it down.
Well, just I don't know how to get it to you, but I could Google it real fast.
Yeah, if you can Google it and then give me the first author and the year.
Okay. Sounds good.
Because this sounds suspiciously familiar to some papers that I put papers in quotations here, that I've seen from people who are basically treating outgroup samples as though they were true representatives of some original genotype, which is not actually appropriate.
Okay. It's in Nature magazine, Ancient Human Genome Sequence of an Extinct Paleo Eskimo.
Is this Flegentov 2019?
Let's see. 11 February 2010. There might be a newer one. I'm sure there's other ones. I think this is where I first read it, though.
Rasmussen?
Okay. Yep. That's what it says. Well, anyway, so there's some population geneticists who looked at the human genome, and they don't publicize what they say, but Dr. Crow said we're inferior to cavemen. Dr. Krondskaloff, no human geneticist doubts that man is degenerating. And Dr. Lynch, even assuming a lower mutation rate, we are degenerating at a 1 to 5% generation. Those are population geneticists, and they just mentioned that in the book that I found.
Yes. So all of those, we're talking about a very specific case under which you have relaxed selection of the human population.
Okay. There's Alex Kronov. I think he's a PhD.
Kondrashov.
Yeah, PhD.
Alexei Kondrashov.
Yeah, he PhD in biology. Why have we not died 100 times over? He said, based on what they found out in the human genome.
Yes, and have you read that paper? Because I have.
Oh, have you? And yeah, I'm sure they come up with justifications why it's wrong.
No,
but and they say there's a big celebration. Okay, go ahead.
The short version is that it would only be relevant under a model that nobody accepts anyway.
Okay.
Could you go into a little more detail about that, CRISPR?
When you get new evidence, you got to change your model.
No, everybody had already rejected the model of hard selection, right? It was well understood that hard selection is not an accurate representation of the way populations actually work. And so it's only in that type of selection regime that you get this kind of problem.
Okay. I got another one. This is the title of it is Sweeping Gene Survey Reveals New Facets of Evolution, May 28th, 2018 by Marlo Hood. You know Marlo? Is it a male or a female? I don't know. Anyway, he says what they did basically is they looked at people around the world and took samples from all around the world, and 500 the house sparrows from around the world and sandpipers, and looked at their genomes and found basically that that they all had the same amount of genetic diversity, and so that pointed to first that it showed that there's a clear delineation between species, and secondly has almost all species being created at the same time, and that's in the article.
Yeah, actually, that's not what the article says. So again, if you've actually read the original research on this,
Uh-huh.
which again I have, um, the actual point of this is that the species delineation, which is done on the basis of mitochondrial DNA, right? There's a high degree of turnover, and there's a reason for this. Basically, because if you just have random drift throughout mitochondrial lineages, some of them just die out by chance, and that's an expected phenomenon, right? And so if you're delineating species on the basis of mitochondrial grouping, right,
Okay.
then there is a upper limit to how old a species can be. It's not because they didn't have ancestors prior to that point. It's because the most recent ancestor of all extant individuals of that grouping, right, would be around that same point for various reasons of population genetics that I could do the math on at a later point if you're
Well,
interested.
I
But,
draw the conclusion that they were all created at the same time, basically, looking at the same
well,
evidence, and you'll never will. And I know you won't get your PhD if you were to admit anything different. But
except
I'm not
except that
restricted by a degree.
except that um, so I'm drawing this conclusion on the basis of having actually read the research and actually understanding the math. And no offense, but I'm betting you haven't read the research and you don't understand the math. So
Okay.
it's not really the same evidence.
Okay. And let's see, the 2016 Royal Society meeting. Did you ever hear about that?
Uh,
The London
I'm just
Royal
guessing.
Society.
Is this the extended synthesis thing?
This is the one that basically the article says scientists see the obvious, confirmed Darwinianism is broken, and so they basically called a meeting. They said they're the ones who started Darwinian evolution. They started neo-Darwinian evolution. It's time for a neo-Darwinian evolution. They said that just that
Yeah.
this whole
Yeah, this is
mutation and natural selection just doesn't cut it. And I'm
yeah,
sure
this is
they caught hell for it for doing that. But
this
anyway,
is this is the extended synthesis. The consensus opinion is that there is no need for an extended synthesis. Um, and the people who think that there is a need for an extended synthesis don't support young earth creationism, right? So like either way, you're not really helping yourself.
okay, well, I'm just trying to make a case against long age evolution.
Can
And
I ask a question? Because
go
it
ahead.
really sounds like you've been going through a list of references coming to you from Evolution News, and
Oh,
that's not a great source of information.
actually, my favorite one is Evolution News. I think I'm friends with them on Facebook, and they got some interesting thing. But you ever go to phys.org? And I like to
Yes,
get I like to get
except
good articles from there, and of course 90%
I
of them
no,
you would agree with.
those
10%
those are
I
not
really like.
those are not articles. Those are popular level write-ups on the articles.
Yeah,
You need to
yeah,
go to the actual literature.
okay. Well, I'm not working on my PhD, but I think they're fascinating what they have, and
Well, again, I just want to point out that if you're not reading the actual primary literature, then it's not fair to say that you're basing your opinion on the same information that I'm basing my opinion on, right? We're not looking at the same evidence and coming to a different conclusion. I'm looking at the primary literature evidence, and you're looking at secondary sources. That's not equivalent.
okay. I like the articles. Do you ever read the articles at phys.org, or they're below you, I guess?
I sometimes get linked those by other people. I scroll down to the bottom, find the link to the original material, and read that.
The original material is more boring for me.
Well, it's
So
hard to understand as someone who's also, is not at CRISPR's level. I've attempted to read a peer-reviewed paper once. Like I found one that actually had the whole, not just the abstract, but the other stuff. And I went to the methods section and I was lost. Like the thing is, that when it comes to like specialties in science and subfields, there's like skills you develop as a scientist that are unique to scientists and to that field in general, and including like in the context of even the subfields. And
One of them
that
is how to read papers.
yeah, well, that's something they teach anyone who's going to these programs. If you're a scientist, they teach how to read papers, how to critique them, of course. But the problem is a lot of this is not. It's not even transferable from one discipline to another. Like it's really specific to that particular field. So trust me, if you have ever looked beyond an abstract and tried to and you couldn't get wrap your head around it, that's telling you something. It's telling you that you really should have this knowledge if you're going to try and critique it. It's like because you need that knowledge to be able to determine whether you're on the right track or not. If you don't have that knowledge, you just don't have near as much information.
Phys.org,
You could be right. But.
Yeah, phys.org, they kind of make it more understandable. And I guess you have to have trust in the author that he's able to understand it and bring it out in a way that's more accessible. And anyway, I'm happy with that because I'm kind of a generalist, and I like to know a little bit about everything and not dig too deep into anything. Okay, I got another one for you. Do you 1,000 scientists publicly sign dissidence from Darwinianism statement?
Oh, see, now you make me want to weigh on this one. I've dealt with this one a billion times too, but I'll let it be between you guys.
Knock yourself out.
No, Crispr, you go ahead.
In a short statement, they said, we are skeptical of the claims for the ability of random mutation and natural selection to account for the complexity of life. Careful examination of the evidence for Darwinian theory should be encouraged.
Well, let me ask one question. I'll let Crispr handle it. Do you believe you could sign that statement and still believe evolution is an excellent model that explains most of the data?
I think that you have questions.
That's fine, but you could, of course, could be an evolutionist.
You could have questions.
You could be Richard Dawkins and still sign that thing honestly, right?
Yeah. I think that as long as you think that there's problems, I think you could sign it honestly.
Does it say, I'm trying to remember the quote. Does it say there are problems with evolution?
Here was a biologist in our network, worked as a postdoctoral research at Harvard. He recounted how about a quarter of the postdocs he encountered were at least sympathetic to design arguments, but none were willing to acknowledge their support publicly due to the likely repercussions. So we don't really know how many question it, and this was kind of anonymous because these people would be hunted down and they would lose their jobs, and they'd get them jobs breaking rocks or something.
So the statement just says that people are skeptical. Hopefully you're skeptical, right? It's good to be skeptical.
And you're open to the idea of intelligent design.
No, it does not say that. That is not part of the statement. The statement does not say anything. It does not say anything about being sympathetic. This was a
quote from a biologist that was...
Well, if I may, I'm sure there are other people who have views on this, but where the dissent from Darwin says we are skeptical of claims for the ability of random mutation and natural selection to account for the complexity of life. Careful examination of the evidence for Darwinian theory should be encouraged. Well, the second part, I think, if you're a scientist, you agree with. You might wonder, because if you know the source of this, you might be like, okay, what do they really mean? But taking it at face value, well, yes, everyone should carefully examine the evidence. That's kind of
and you should be skeptical of all claims.
Exactly. And the first one, well, honestly, I think we've known for a while that random mutation natural selection is not the only thing there is. It's primary, but it's not the. I mean,
it's not even primary.
Okay, I'm not going to argue with you. So there's plenty of other factors involved in how things evolve that explain the complexity of life. So mutation natural selection alone is not enough. So again, taken literally, I'm skeptical of this. I could sign this very easily, even though I'm, I guess you could call me a devout evolutionist who is unlikely to change his mind anytime soon. So I wonder whether this really, no matter how many people sign it, if everybody signed it, I don't think it would really get you anywhere.
And just to comment on the hounded out of the field comment, I actually knew a signatory to this list who retired at a reasonably old age from his posting, who was actually a moderately serious ID proponent. And he had an official university affiliation, right? So they don't get hounded out. That's what tenure's for. You can say what you want.
And so when did mutations start becoming an issue, like from the primordial soup on?
What do you mean by issue?
I don't think that's an answerable question at that point.
Well, the evolutionary theory needs a high rate of beneficial mutations, and it's the only actual thing that actually changes the genome. You got gene drift and all this other stuff, but as far as making genetic changes, mutations are the only thing. And so, I mean, supposedly there's some real simple life forms that started off, and the dog ate the homework, so we don't have any evidence of those. But even the most basic in the pre-Cambrian level are incredibly complex. And were there mutations changing it at that point?
We have stromatolites, which are single-celled. We have actually substantial evidence of single-cell organism activity, well predating any evidence of any advanced organisms.
Okay, they didn't have DNA or what?
No, of course they had DNA.
Okay. And so they were pretty complicated then.
All right, guys, in a couple of minutes,
cells.
if I may, in a couple of minutes, we're going to start doing some Q&A. We're going to bring in some whatever people come up with in the outside chat. Go ahead and continue for a minute. I'm going to give people time to go and come up with their questions. They have to write them into the outside chat. So continue on for a few minutes, and then I'll cut us off, and we'll start Q&A.
All right.
Yeah, so the point I was trying to make there is that you do have evidence of single-celled organisms long predating the Cambrian strata. So I just wanted to make that correction.
Okay. And but there weren't really simple ones. They were pretty complex. And then in the
they were single cells.
When you get to single cells, but when you get to the Cambrian level, now all of a sudden we got the trilobites with eyes and brains and everything. And within a period of 5 million years, do you think that
you're skipping the Ediacaran?
Okay.
You're skipping the Ediacaran biota.
Okay, just from the pre-Cambrian level to the Cambrian level is supposed to be 5 million years. There might have been something lower at the Cambrian level, or where is that?
No, it from pre-Cambrian is everything before the Cambrian, right? So that's a pretty big chunk of time.
It's a very big chunk of time, yeah.
Right. And so the Cambrian explosion, quote unquote, is 50 million years at least, and the Ediacaran is another 100 or 150 million years or so, right? So this is a huge stretch of time.
Not really any evidence of transitional forms.
You have evidence of single-celled organisms,
yes,
and then multi-celled organisms, and then you have evidence of more advanced organisms that finally have hard body parts that tend to fossilize. And here's the thing, even if I were to give you that we have no understanding of anything prior to the Cambrian explosion, after that, we have fantastically accurate evidence of all of the major transitions of life.
There are some problems with the fossil records. Would you agree?
Define problems.
Oh, just with the long age, and some of them are upside down, and some of them aren't in the right order, and some are missing some, and there's fish and the sea plants all the way up to some of the highest levels, and just as evidence for evolution, what other evidence do you have besides the fossil record if that was proven wrong?
I don't need the fossil record. I can do it on genetics alone.
Similar design?
No, the genetics.
Similar design?
No, it's not design, it's genetics.
Genetics? Similar genetic design?
So there's no reason to assume that the majority of the completely non-functional genome is designed, right? If it is designed, then it's necessarily designed to look like it evolved.
Well,
So your options, your only
well,
logically possible options are evolution or a designer who wants you to think that evolution is true. That's it.
well, I don't like all the ideas of junk DNA and 98% junk, but I think we're learning more and more, and that the 98% is basically a myth now, and I think
It's not.
categorized as a myth, and the cell does a lot more things, like they communicate with other cells, and the DNA, I think, is the program for all the mechanisms that go on in the cell, and I think the DNA is incredibly important besides just coding for proteins, and so
Yeah, well, sure.
different
But
idea.
all of those functions occur around 10 to 15% of the genome.
Yes, okay, it's getting bigger, but I think it's going to
It's
keep
not.
getting bigger, and
It's not.
just as we learn more about it, and you got to be
You're
open to new
okay.
evidence.
So you're saying this on the basis of what evidence?
Well, other researchers have gone into it, and of course they get in trouble, and they're going to lose their jobs unless they're working for Answers in Genesis or something. So anyway,
So that's a claim with no evidence.
well, that's what happens to people, and then you're going to work in. You're going to be working as a biologist. You're going to be working on government grants, and so they grant about 12,000 grants every year out of 50,000 applications, and so you better have something that the NSF wants you to have. If you just like put in for a grant for proving that there's more information happening inside the DNA, they're not going to give you a grant, because it just doesn't go along with the
thing is, right? Hold on.
Okay.
The thing is, that kind of information would be, if true, be extremely valuable for fields like cancer research and translational medicine. Okay, and you cannot stop that kind of research from happening if people think it will work. Which means the fact that it's not happening means people don't think it will work. This is just like, why don't oil companies use creationist geology to find oil? It's because it doesn't work.
No,
it's
I don't
the
know.
Same thing. It's
I
the
don't know
same
that.
thing with creationist genetics. It doesn't work.
No,
Well,
I don't think that's true.
Oh, I can corroborate the one about finding oil. There's petroleum geology is used by large corporations to find oil, and all the software that they use requires things like the age of the Earth, the absolute ages, the ones that you would disagree with. They use those as an input to try and find oil in places where they haven't done a lot of digging. And there's only one company in the world that uses young Earth ages, and they found no oil. The other ones apparently are popular enough that companies like Exxon then buy their software because it works.
I think you just have to figure out how oil forms, and then figure out where it's most likely to happen. And
Right.
It doesn't really have to be involved in the age of the Earth, I don't think.
Well, actually, it does,
The
because
thing is,
time
doesn't-
and pressure
hold on,
is important
hold
to
on,
oil formation. Go ahead.
And then Go ahead.
doesn't necessarily, but as it happens, it does, right? So in practical fact, right, the best way to find oil is to understand that it is formed on the basis of an old Earth geology. And if you
I
don't use those assumptions, you don't find oil.
Okay.
So go find oil.
Okay, here's an assumption. I assume that oil forms rapidly, and I say go back to some dry wells and see if you find oil in them. If it's in the right spot and they have the right hydrocarbons and the
Okay,
right.
if I may, the point of the software is to find oil when they've done very little surveying.
Okay.
So you're basically in an area that you really don't know a lot about. This software can be used to help you find oil. But the thing is, it does require the absolute ages as an input. It's part of the formula. If you don't put that in, that doesn't work at all.
And so actually I have a friend that works in oil fields, and they found old oil wells that were pumped dry, and now they over half full again. So I believe
that's a whole other issue. Anyway, we're kind of getting a little off the topic here, and Unfortunately, I don't see a whole lot of good questions from outside. Everyone's having fun yelling at each other. They're not really. There's a couple people that are having a long-ended conversation, kind of yelling at each other, and so we don't have a whole lot of questions. Hopefully, there's some more we'll pour through. Let me try and find a couple that I liked. It's gonna be hard for our main moderator Drag to point them out. So the first one is for you, Cameron. By the way, there are a couple creationists out there. I'm trying to get questions for CRISPR. So far, they can't think of any. I think we've converted them. I think they're all evolutionists. They now deny God and the Holy Spirit, and they're gonna go. They hate church now, and yeah, no, that's not really a thing. All right, so
I gotta stop you right there. Working in the sciences, in the natural sciences, I know a large number of people, and many of them are devoutly religious, and they're also evolutionists. These two things are not contradictory. I just couldn't let that stand. I'm sorry.
Right. Okay. Well, I was just joking, but fine. Correct me, jeez, you big jerk. All right. Well, anyway, so Cameron, question is for you. Is intelligent design falsifiable? Is there anything we could find that would prove that intelligent design is false?
Well, I first of all, I don't like the big issue of things being falsifiable because, for example, can science prove God? No. Can he prove the inexistence of God? No. But anyway, intelligent design, I would say that you find design, functional design, that points to an intelligence, and that points to a purpose, which would draw the conclusion that it was from a designer. And so if we had life forming from non-life in ponds, I think that would disprove the need for an intelligent designer like Darwin believed. And how would it be falsifiable beyond that? I really don't think I don't think it's possible to falsifiable. It'd be more looking at the evidence and coming to the conclusion that this didn't come here by time plus chance. This came here by an intelligent designer. And the job would be try to find out who that is or what that is.
All right. Well, I think the point behind the question is that if you don't know what would show that you're wrong, it's how would you ever know that you're right? If there's no because proving yourself wrong is kind of how science tests ideas.
And so,
you need to go like, well,
functional design coming together by chance, it would be the only way it'd be falsifiable, I believe.
Well, that's not falsifiable. That's simply an alternative. I mean, of course there are alternatives, but it'd be very easy. I mean, could you imagine if evolutionists said, well, evolution is right because we have not proven that this other explanation. Well, that's just an argument from ignorance. That's just saying that we don't have an alternative, so this one's right, and that's a really bad reasoning. Anyway, CRISPR, what do you think?
Well, I mean, firstly, science doesn't prove things. It's not in that business, right? That's not the point of science. So I just want to make that correction right there.
Well, we can use proof in the we can use proof in the colloquial sense, though.
But I'd rather we not.
Well, I'd say evolution would be falsifiable if you prove mutations couldn't do the kind of change that they just cracked up to be. And if the fossil record, maybe if you prove the fossil record wasn't long ages, then that would disprove evolution too, I believe.
Right. So I think I actually did provide supporting evidence that mutations work in that way. And we've not heard anything to the contrary of long ages in terms of the fossil record. But anyway, with respect to ID, the important question is, regardless of whether or not you're trying to falsify a designer, what is your statistical test? That's the real question. How are you showing that whatever you found is significantly different from whatever you expected or didn't expect to find on the basis of whatever your hypothesis is? If you can't put forward that, you've got nothing.
Well, I would say a finely tuned universe that is set up for life, which a lot of cosmologists agree with. And anyway, looking on life itself and what we have down here on Earth and how amazing Earth is and how amazing the sun is and how amazing the solar system
Can you
is.
see how this isn't an answer, though? Right. So my question is, we all agree, right, that, you know, the universe is pretty neat, right? But the question is it neater than you would expect in a statistically significant fashion? Neater than what random and stochastic and natural processes would provide?
Yeah, and so
That's
anyway,
the
well,
question. So
well,
demonstrate
Stephen
the
Hawking,
significance.
okay, Stephen Hawking all came
No.
together by
You're about to throw a
gravity.
name. You're throwing out a name. Don't throw out names. Give me math.
Okay.
Give me math that says that it's
Okay.
significantly different than expected.
Okay, I just think logic shows that if there was an explosion, you're not going to get an orderly, well-organized result. And I was in the Army, and I blew up things. Big Bang.
Who said it?
Big Bang.
Bang's not an explosion.
Is it expansion?
It is an expansion. It's not an
Okay.
explosion.
A rapid expansion of space-time.
Yeah,
I
and
mean,
so I don't think you can get order from a rapid expansion.
Well, if I may, the problem with trying to discuss, the densest, hottest point our universe has ever been in, it requires a phys.org article is just not going to cut it. You need to understand quantum electrodynamics, quantum chromodynamics, vector calculus, and algebra. If you don't have the skills to understand that kind of math, if you can't fill a whiteboard like a Big Bang Theory episode, it's really good to stay away from something.
Well, I like to Google things like
Google's fine. Google is fine. Google is fun.
Problems with the theory of relativity.
If I may, there's one problem though, and that is the Big Bang. If you go back all that far, if you go back to the supposed singularity that we don't think really happened, the math breaks down. If you're getting to the point where the math breaks down, you're already at the end of our knowledge of things. We're still working to try and fill those gaps and fill things up. So if you don't understand the math at all, or the fact that math breaks down, or really anything, and if you still think it's an explosion, you really need to get that stuff fixed first before you start asking, and maybe then you can ask better questions.
I think
there's a larger problem, right? So in all these times when you've done Google searches for problems with relativity or problems with evolution,
quantum,
have you ever done a search for solutions to problems with?
Oh, you'll find that. You'll find articles for that when you do that.
Because otherwise, you're really biasing your search pattern, aren't you?
Well, it's to be open-minded. You already know that this is settled science, and I just like to look and see if there's any other evidence. And the whole idea that mass created energy, I don't believe. I believe energy comes from an outside source and not from mass. And so much for the theory of relativity and quantum.
Okay, well that's way off of our original topic of mutations.
Well, let's talk about design in the cell and design.
Although I do actually plan, if we can, I want to have you back. We have a physicist in the room. We discuss electric universe and plasma theory.
Yeah, okay.
That would be fun, but that will be for another day. So we're gonna.
Yeah, okay.
I'm actually gonna make up a question.
Design in the DNA in the cell.
Right. I'm going to make up a question for CRISPR here because so far the creationists out there are too busy yelling at everyone else. They don't seem to be even listening to the show. So this is from Mashimi. I'm going to invent this one. So CRISPR, is adaptation different than from evolution? CRISPR, did you catch that? Crap, I think we lost him.
Oh no, that's a tough question.
Yeah,
I think adaptation could be.
This guy believes adaptation isn't evolution.
Intelligent.
Yeah, well, he believes that he's a creationist. He also believes that that we were designed. However, he says adaptation isn't evolution. What do you think about that, CRISPR? Is that true? And CRISPR, unfortunately, may have been.
Well, let me tell you my idea.
Sure. Well, go ahead, Jeremy.
model,
You go first.
and my model is that that we all started as a different type, and so there's like 300 different types of dogs, but I think it all started as a wolf-like creature that had all the genetics for all different types, and then I think mutations gave us the speciation, and by limiting information that was passed off, some just became small dogs, and
well, I actually have a small problem with that because you have a problem with the idea of positive mutations for the most part, right?
yes, I didn't like his explanation.
Well, unfortunately, that causes a problem for you if you believe Noah's flood happened around 4,400 years ago, because according to most creationists, there was a dog kind, and from this dog kind, whatever form it took, from it we got teacup dogs and Great Danes and poodles, and even further we have different types of wolves and huskies, and even foxes. Now, if you look at a
they're
fox,
the same kind, and they can make,
they say they're the same kind.
together.
However, if you look at the difference, in mutations, positive and negative, they're huge. There's the difference between a teacup dog and a fox is remarkable. Now, I think if we were to I were to claim that this took 20 million years, like, oh no, it's not possible. That's too many mutations. But suddenly we say it take only 4,000 years, and now you think, well, that's actually fine in that span of time. But I thought it was the other way around. It takes so much longer to get that level of mutations, but it looks like it only takes 4,000 years. And suddenly, from 4,000 years, we go from the original dog kind to foxes and wolves and teacups and poodles. We get this remarkable amount of variation and a huge rise in positive mutations from the original.
Teacups probably wouldn't survive if it wasn't for humans. Go ahead.
So interestingly, it appears that my audio setup is not intelligently designed either. So anyway,
we lost you for a moment, but you're back. So the question is, do you got the question?
I got the question. I heard it initially. It was just my mic decided it was going to be hardware muted, and then I couldn't get it out.
It developed a negative mutation.
Yes. So adaptation isn't evolution. That's an interesting thought. So adaptation is an effect of selection, which is part of evolution. However, most of evolution is actually non-adaptive. This is actually something that people have a lot of trouble with in the modern world. People like to look at the adaptive processes because they're the obvious and they're exciting ones, right? But most of the differences tend to be actually non-adaptive. And then when environments change around the species that already exist there, then suddenly that opens the opportunity for existing variation, non-adaptive variation, to spread throughout the population. I don't know if that's actually answering the question, but it's a very important point.
Yeah, I'm looking for another good question out there. There's just not a lot of them. All right, here we go. This one is for you, Cameron. What are some things that are definitely not designed?
What are things that are definitely not designed? I would say, you see, rocks. Things that don't have any functionality to them. Rocks kind of have a story of how they got there and stuff, and I'm interested in geology, but
so God didn't make rocks. If you see a rock, God didn't make that.
I wouldn't necessarily come to that conclusion based on the evidence, and so but what did John Hutton? He had Plutonian thoughts, theory. It was that Pluto was at the center of the Earth, making all the rocks, God Pluto, and so I think that God made them, but I don't know that would be necessarily proof that I'd hold out compared to DNA for intelligent design.
Okay, Crispr, anything to add?
No, I was I was trying to respond to something to drag in the side, so I think I may have missed a bit of it.
That's okay. It really was just aimed almost entirely at Cameron. Well, let me just go ahead and get your final thoughts. We're actually already into overtime. We're usually trying to stick to an hour, but we went a little over. Cameron, you go ahead. What are your final thoughts?
Well, I just think that upon myself examining and analysis of the evidence that gene mutation are the raw material for the evolutionary process, and I think just thinking that they could do all the things that a cell does, and just the mechanisms that are involved and that fix mutations, they came from mutations, and just that just because of the complexity of the cells and then the human body, the eye, the brain, everything just coming from mutations. Just I don't believe that they're powerful enough, and I know evolution cannot succeed without mutations, and evolution cannot succeed without them, and evolution is just not possible, and it points to an intelligent designer that made plants, animals, and all the complexity that we see, and mutations, with the majority of them being harmful, they just would anyone that are functional just wouldn't be able to survive, and just could not do everything that they think evolution has that they need to do. That's my talk. Crisp gone again?
No, I'm
Yeah. All right. Thank you.
I was waiting to be introduced.
Thanks again, Cameron. And yeah, Crispr, go ahead.
Yeah, so, I mean, mutations demonstrably occur. Mutations are demonstrably heritable. Mutations demonstrably have effect on fitness. Some of these effects demonstrably, depending on environment, can be beneficial. And that's all you need. That's the whole game right there. So I don't know what else needs to be said.
All right, then.
All right. Chris, thank you for telling me
and
about the things
thank
I
you-
enjoyed.
thank you
Good luck
for
in your
doing
career.
this.
Yeah, thank you guys both for coming on. Really appreciate it. Well, I mean, just hey, for everyone out there in the outside chat, thank you guys all for joining us. We're sorry about the start time getting a little mixed up. It was not my fault. We're gonna blame CRISPR for this. It's all CRISPR's fault somehow. I also want to thank everybody who helped to get the show off the ground and get it running. To all you guys out there, I will name you by name later. Apparently, it's a very, very long list, and I don't have time to really read it all out. In fact, we think we might be missing a few names. So until we get the full list, thank you everyone who helped to get the show going, and just thank you so much. That's all for us now. Goodbye and farewell.
See you next time. And if you want to check out my book, it's